It's my great pleasure to be here to talk about this regenerative medicine and impact on the healthcare. I'm very happy and appreciate the Professor Mishra to invite me give a talk about how to improve the healthcare using stem cell technology. And today, this presentation is very special for how we can improve the healthcare using the stem cell technology. And this stem cell technology is a little bit different from other conventional stem cell treatments. So the topic is regenerative medicine's impact on healthcare. So I think everybody heard about stem cells because it's booming now. But stem cell treatment is a broad name, very, very broad name. And its application from different diseases is different methods, the isolation, manipulation of stem cells, injections, there are a lot of long procedures. But we prefer to activate the endogenous stem cells. The endogenous stem cells, how I can do the next? Yeah. So, if anybody has questions during my talk, please interact me during my lecture, then it will be much better. So, if you have any questions or any doubts or anything, you can interact me during my lecture. So, let's give me some historical backgrounds about the stem cells. In 1958, the Professor Thomas, he was the first person, he was a doctor, and the first person taking the bone marrow transplantations to the leukemia patients. In 1958, in that time there is no stem cell term. And then when he injects the bone marrow to the leukemia patient, most of the patients are cured. And because of this simple technology, with great impact for the healthcare for leukemia children, he got Nobel Prize in 1990. That time there is no stem cells, it's just bone marrow transplantations. Because bone marrow is a house of the stem cell, endogenous stem cells. And then next is embryonic stem cells. Embryonic stem cells, it's that embryonic stem cells can make any type of stem cells, but it's associated with ethical problems, because to take the embryo, stem cell from embryo, and you making the different type of cells or injection, it's making a lot of ethical problems, lot of regulatory issue globally. So it is very good, very high potential technology, powerful for to cure many diseases, but unfortunately it has ethical problems. And the next one is recently Shinya Yamanaka and Sergio Gordon got Nobel Prize because of the IPS technology, because it's called artificial embryonic stem cells, because they make it from skin to artificial embryonic stem cells called iPS cells, and it's totally equivalent to the embryonic stem cells. This is the funny and interesting things is John Gordon in 1958, he first he proved that mature cell can be converted to stem cells using the frog models. And that in 1962, Shinya Yamanaka is a birth year. And he developed the IPS technology. He proved this concept of the Sergio Gordon in 2007. See the difference, 1958 until 2007 is a long time, more than 50 years. And then he developed and he proved that all mature cells can go back to the stem cells, and we can create the artificial embryonic stem cells using the skin cells to any cells. So this is, and that's why they both got Nobel Prize in 2012. So So just imagine the 50 years he developed the concept. This is sometimes the science, if you go to the science history, sometimes you can see the dark period of science. If you develop something, sometimes it's very difficult to believe. And many Nobel discoveries are like this. I met many Nobel laureates, and they said, okay, somebody like nitric oxide, like 9 years, who developed the nitric oxide, that discovered the nitric oxide synthesis in the body, and 8 years nobody believes. So all Nobel discoveries are like this. So it's not believable sometimes, but in future they got Nobel Prize because it's a science. So the first is endogenous stem cells, and this we are working a lot. We have treated more than 2,000 patients, and then induced pluripotent stem cells. The endogenous stem cells present in our all organs, and it's also called the progenitor cells. You can see the repair. Can you do a pointer? Repair and regeneration. So repair is associated with some abnormal scar or something, and when the stem cells, endogenous stem cells, participate more often, it regenerates the tissue, cells, organs. So the key is how to activate the endogenous stem cells inside the body. Then you can regenerate any cells, tissue, and organs. And the right one is iPS cells. As I mentioned, this is novel discoveries. If you can give skin cells, after 2 months, you can create a liver cells, you can create a heart cells, you can create any cells. But this is the procedure to create skin cell to heart cell, liver cells, or any cells. We use, there is some oncogene factors. So still it is a limitations of the cancer initiation or cancer creations. So that's why still it is not in clinical trial, but it's very extremely good for in vitro clinical trials, for drug testing, preclinical research, and so on. In Japan, that's from iPS cells, they use clinical trials for patients for macular degenerations, but the patients are quite good and they improved a lot, but still they are in process, and they believe that it will become in future clinical trials. They are working hard for move forward to the clinical trials, and I believe it will be sooner or later it will become to the clinical trials. So if this will be solved, then it will be great for healthcare, and so that you can take a skin cells and you can make whatever you cell want within 2 months, or you can create your own cells. Suppose you are healthy, you are everything, you are healthy and you don't have any health problems, and in future you don't know when you will be ill, when you will be sick, when you have any heart problem, liver problem, or kidney problem. So you can give your skin and make it kidney cells or liver cells or heart cells and make it advance like 20-30 million cells and keep it preservatives. And then when you any problems directly, you can. This is the future prospects of the iPS technology, and I believe it will be work sooner or later. But transplantations, everybody, like all clinical centers, suffer from limitation of the organ supply. So it is it's good options, but it is the main limitation is the patients cannot get treated because of the lack of organ donors. The concept is the human life. Human life is 70 to 80 years. In some countries or in some community, more than hundreds. If you can see the lifespan of the functional cell inside the body, let's say in liver, hepatocyte cells, 3 months. And the questions coming that when the hepatocytes die, now where the new cells come from? The answer is the local stem cells. For example, in heart, before 2009, nobody believed that heart has a local stem cells. When the paper published in Science in 2009, not so long ago, just recent, let's say in 10 years, and then researcher and scientific all that they believe that heart also has a stem cells, local stem cells, and they can repair the heart. And right now also we have detected some stem cells in human heart. I'm going to show them some slides. So the thing is that all organs they are all local stem cells. It's very important to understand, and it's very important to detect the stem cells, the location of the local stem cells, where they're located, because main key is to activate the local stem cells when the organ is in problems. So it's very important to know the location of the local stem cells. In liver is almost located, it is known, and in heart also almost known where is the local stem cell located. In others are also a lot of investigation had done where is the local stem cell presence and how it participates for organ regenerations. The very important points is that's the power of local stem cells. I told about this bone marrow stem cells, and local stem cell is different. The bone marrow stem cells and local stem cells. Bone marrow stem cells present in the bone marrow. When any organs has injured or aged in some problems, they try to mobilize, so the stem cells can migrate from bone marrow to the other organs, because our body know how to regenerate the organs. But if you can compare the bone marrow and local stem cells, the most highest regenerative power is the local stem cells. It's also called local progenitor cells, and basically they are silent and sleeping. The stem cells has, like local stem cells, bone marrow, if they are not properly there is not properly a microenvironment of these local organs, the stem cells can go to the oncogen initiation. This is the major limitation of the stem cell treatments. It is shown that if you can compare the bone marrow stem cells and local stem cells, then local stem cells they don't go for cancer initiations, but bone marrow stem cells can go to the cancer initiations upon some abnormal conditions. So if you can compare the local stem cells with bone marrow, then local stem cell is much superior than the bone marrow. In globally, you can see the all stem cell treatments everywhere. They try to isolate the local stem cells, the bone marrow stem cells, and they're getting 2-3 million cells, and then they try to manipulate 40-50 million cells, and then try to inject. But the problem is the conventional isolation of the bone marrow and magnification up to 40-50 million bone marrow stem cells and inject back to the patient, even in allogenic, still it is associated with high inflammations. Some patients are dying, and lot of immunocomplication problem has been shown from among a lot of stem cell clinical trial center. So if I say the bone marrow stem cell transplantations still has a major limitations of the inflammations, then you can ask me how we can isolate the local stem cells. The local stem cells, as I mentioned, it is very low number, and it's not easy to isolate the local stem cells, but there is local stem cells somewhere in each organs, and they can activate when there is emergency. As I mentioned, that when the lymphocyte cell die or cardiomyocyte cell die, then our body know, okay, the cells are die now. We need a new cells because we have to make organ regenerations. They have automatic control that locally. Okay, now cells are dying and new cells coming from, and then regenerate. We don't know when the hepatocyte die and something. So we live 70 to 80 years, and we try to detect the local stem cells where it is and how we activate it in health conditions, how it is present in health conditions, and how it is activations happens upon normal injury, and how it activations happens in the major problems, and why if you have big problem, big complications of the heart or any organs, then why the local stem cells cannot participate for whole regenerations. Yes, the local stem cells try to regenerate in any conditions, but they are not able to regenerate in the major complications. So that's why I'm here today, that how to activate the local stem cells. If you have major problem, let's say in liver, if you or any heart, if you major problems in heart or at that time local stem cells cannot able to regenerate, but they are there. This is important to know that local stem cells are there. In that condition, we need to think. Let's try, if somebody is dying, let's try to activate these local stem cells. If we can success, then this can be practiced it can be applicable to the other patients. So we have some experience in the diabetic food. So I'm going to share in next few minutes. So local stem cells has the highest regenerative power, and it can participate in any case. But we need to find out. We need to find solutions how to activate these local stem cells. And there is another important point is it is like personalized therapy. Like it's not standard method that's okay, you have this procedure and this no. Because person to person is vary. Like each person has their own lifestyles and their own endogenous stem cell system. Like we know everybody that's an immune system or any digestive system. We know like these systems, but we don't know about the endogenous stem cell system, because every organ has their own local stem cells present somewhere on these organs, and we name that endogenous stem cell systems, and this is called, we name these local stem cells silent and sleeping stem cells. And generally, most of the times they're silent and sleeping. If they only they wake up when they need it, means when they're injured or aged or some minor injury or major injury, they can participate. I'm telling about in the major cases, if it is the major complications, you try to activate the stem cells, the local stem cells. And I'm going to show some cases. Then it can be success. Because if local stem cells can activate it, they can create the entire organs. But we need to find out which recombinant proteins and which hormones, or we need to make a proper combinations. So now what stem cell research are doing, so you can find any type of stem cells. You can buy, you can request from any university, okay, please give me this type of stem cells, and you can develop in vitro models. Suppose I can give you or heart progenitor cells, then your target is to treat the heart patients or any organs. Then you can try to do in vitro experiment. Okay, this is a progenitor cell. This is not a cell line. This is progenitor cells, primary progenitor cell line. And then you can make it some combinations. You can see some growth factors, and if you try to activate, stem cells proliferated, and that's fine. Okay, then this combination is the perfect combinations. Maybe then you can apply those. Most of the recombinant proteins you don't have any problem to make it for permissions, because it's already a lot of growth factors is already approved for clinical injections. So if you can combine some growth factors, recombinant proteins, and to activate the stem cells in vitro model, and we found this, let's say in cardiomyocytes, Okay, you develop some formulations, and then you activate, you can make a lot of cells in, and then maybe this combinations you can inject to the heart. If some patient is dying, about to die, you can try. Then then the stem cells can activate, that they can create a new cardiomyocytes, so one patient can survive. And we have cases on skin cases like this. So this is the point, that now we have to target the local stem cells. And even bone marrow can also participate. We don't know, because our body knows how to regenerate better. So our body knows much better how to repair regenerate cells and tissue and organs. So let's consider our body is a laboratory, our body is a culture plants, and our body is everything. Stem cells are there, and we don't need to take out the stem cell from body. And we need to know that where it is the located stem cells. But we have to find out the special formulation for those patients. So it's not like universal. So some formulations can it's not a standard. So we can make a also personalized therapy. So we need to analyze the patient, the profiles, certain profiles. Accordingly, we can make this combination. Then we can try. So this is purely personalized therapy at the point of care. So the line is very simple, but if we will follow this line, then we can treat a lot of patients. Things that our body knows much better how to repair and regenerate cells or tissue organs. We need to supplement. We have to supply some supplementary factors, because stem cells are there. Once they come out from outside the body, there are a lot of changes. So let them inside, and we need to think about how to activate them locally inside the organs, injured or any complications. We name this. And bone marrow stem cells, as I mentioned, it is still working, still in some cases, and embryonic stem cells have problems with ethical problems. And stem cell mobilization. So stem cells in bone marrow, stem cells are ready to live. And there is a lot of researchers that develop mobilization agents. So if you inject the mobilization agent, then it releases the stem cells, and then move to the injured organs for regenerations. Like as I mentioned, our body knows we have very natural type of regenerations, very high power. Billions of stem cells come to the injured organs per second. It's like faster than the rocket. They're moving very fast when we during organ regenerations. So why we should not consider them, let them inside, and how to give some support to activate them. And this is the stem cells. So third choice is, you can see a lot of stem cell center, they use simple concept. That's taking the bone marrow and using some GMP, GCP culture medium and making some millions, because we need to make it 40 more cells, and we cannot get 40, 50 millions, lot of cells from bone marrow. We can get 1, 2, and then we can make it. This is associated with complications, inflammation, lot of issue. So you need to make a GMP, GCP procedures, a lot of things, and once cells come out, they make inflamed, and a lot of problems with third choice. Second is, you see that if some cases that we cannot activate the stem cells inside the body, then you take the stem cells from outside body, we do, and we don't want to keep them not more than 30 minutes. So we develop this technology. First is the most choice, and we often use this one. No isolations, activation of sleeping, and silence endogenous cell inside the body locally. And how? The point is that questions how we can. It's not easy to activate these sleepy stem cells. If we target skin, we need to know where it is located in the skins or any organs, in heart or kidney. Still, it is a lot of result, even including us, getting very successful results for activation of local stem cells. This is one thing that's allogenic. It is a lot of problem. We make a tables here. You see, like even in allogenic autologous stem cell transplantations, almost all of them need the antibodies and has the complications. Some patients also die. So we make some review work what's going on globally. So we are now in phase 2 clinical trials for skin. So when we activate the local skin stem cells and we don't see any scar, it means that local stem cells, progenitor cells, is the main key for regenerations, because they know, because they used to repair the cell, tissue, organs throughout our life. So they know better, and we activate them. Then it's fine. Then everything and repair. So sometimes skin repair with something. If you get injured, then it make a scar. But it's a repair always associated with scar, and scar is not a perfect regeneration if it is a skin or any others. So if we target the local stem cells, I hope that's scar-free, like no fibrosis. And no fibrosis is the big problems. So we can control the fibrosis like scar. This is also published in Drug Discovery Today. We have back for stem cell, only for human, stem cell treatments for diabetic foot. You can see how many number of cells they inject and all these things. And in small wound, we don't go to the hospitals. And if we try to make a small injury, and then you see what's going. Any questions? So if you have a small injury, then generally we don't go to the hospitals, and we try to know what's going on the small wound, so it repair completely. And if in cases of the big, and it's a deficient of the cytokines, growth factors, and then it got infections and so on, and then finally it make a big problems, in particular in diabetics. And if you apply some of the growth factors to activate the stem cells, then we can repair the diabetic wound. And it's not only the one factors. We can make a special combination of the growth factor for different patients. Depends upon the patient's health conditions, depends upon the cytokines profiles of the patient, age, lot of things. So just like when you go to make a shirt for the tailor, we need to make okay, this will be fit or this will not fit. And so first concept is that we have to target the local stem cells. Second thing, we know where it is, and then how. If we formulated something, some growth factor cytokines, then how we apply there? Injections or how? So we need to put some supplementary factors on local side of stem cells. And this is also another key step. So it is called like a slow release concept. Slow release concept means if you inject, or if you suppose you develop some growth factor cytokines and you try to inject every day or every 3 days, it does not work. We test it in an animal model. So because excess is not good. So in the same principle also, this is if you inject, if you even develop an in vitro model, you optimize everything, and then if you inject it does not work, even it can problems. What do we think and what do we prove? That let's make a hydrogel and put the all growth factors inside the hard gel and put aside on this side stem cells, and stem cell knows when they want from them. It's called slow release. So they speak each other. So we have excellent network inside our body. If we mimic some molecules, then they can speak each other. Okay, now I need it. Okay, release. Okay, now done. Don't release more. Deep slow release concept is very useful and it's very important point for successful treatments. So that's why we develop the hydrogel, put the growth factors inside the hydrogel and putting on the local side of the wound, and then it release. And it's not like then we wait. And you can say, ask how many days we should wait. We don't know. We generally wait 3 weeks. If not, then we can give again. But most of the cases, when we wait and like within 2 weeks, we see the new skin coming up, which is totally new. Nobody has done till yet, we believe, best of our knowledge. Then when you see the new skin coming up, then stem cells actively participate, but it's very slow. Like for one diabetic student, it takes 9 months. It still is okay. So you see, if you put a saline, it could degrade, and we develop this hydrogel. It's constantly stable up to 30 days. So around 3 weeks. So stable the hydrogel. We put this. We can make it any growth factor cytokines. Depends upon the persons. Develop it here and keep there, and then when they need it, they can take it. Very simple, but it's not so easy to develop the cytokines and growth factor. Let's say here is also we make it for cosmetics. So skin, as I mentioned, stem cells everywhere. So if you somebody for anti-aging concept, so if it's stem cell also here, and if we apply, and then it can be observations. This before and after, we have treated like such cases more than 100. And you see the local stem cells here, then they prepare and they make more activations when they put in little injury. So any skin injury make an injury means they are wake up conditions. They try to wake up, they try to activate, but not able to. And putting of them some external factors, they'll be happy. They happily welcome such factors and they participate in regenerations. This is the cases which we have in Germany. My boss is a surgeon, and then the patient is the diabetic one, and he's not ready to cut his legs. And he Googled, I'm telling about in 2007, and then he Googled and found that okay, that we are at that time we are doing for some research work. And then he's not ready to cut legs, but he's ready to do any treatments. In Germany, if any doctor and physicians can treat 9 patients up without any permissions. And if he is successful for the 9 patients, if he make a great treatments or is successful, then you can go to the government, you can ask them, then give me some millions euro, we make an extension. And this has happened in our cases. We got 3 million euro for phase 1 clinical trials. And you can see in these cases, we also published, like we submitted in Lancet kept it several months, but finally they said no space. So from there to here takes 9 months, but still activated. And the patient is completely cured and go to back to the old world. So this is the new discovery for us, and we were very excited about this, from this to this skin. And then when the cosmetic people from US and France, when they came to know that if we can make it such skin to such skin, then why we cannot make a skin creams? Finally, we developed a skin cream also. So some slides that in deep second degree in burns generally make it multi organ failure. But you see here, in 10 days it recovered, and finally in 6 months you don't see any little scar. In generally deep second degree burns, it is like some scar, but you don't see any scar. We used some profiles of our treatments that everything is normal according to the German regulations. And you see here Then it's in German. The slides in the last one, Tag 12, mean 2 weeks. We found neurons, we found new blood vessels, we found a lot of different things in when we make treatment. So we make part of the clinical trials. So this is finally we develop a skin creams because it is launched in 2018 in Valentine's Day. It's a premium product and we named it to my boss now. And he's my boss. And a lot of Hollywood actors from US, they came to us and we make them more beautiful skin, and because they are the investor, because they wanted to put in money and they want, before putting money to the company, they have to try it themselves. So they are quite happy.
Do you guys inject just stem cell or along with the cytokines?
We don't inject stem cells, we just make a special combination of growth factors.
Growth factors?
Yes, and then we try to putting.
How do you decide that growth factors?
Yeah, it's a big deal. It's the main key point. It's not easy, and it's the main theme of these things. So we need to check your profiles, blood profiles, everything, some screening procedure. And we see the profile, okay, you have such profiles and these things. And then we developed our own platforms, that this type of things, then we need to make this. If we found a new things, then we can make new. So it takes 2 to 3 days to decide, at least. And the good point is that we don't see any scar and we have almost 100% successful.
So, looking at the pictures, it looks like the wound is due to some kind of infection, right?
It's a diabetic?
Wound in the picture?
Yes.
It's a diabetic?
Yeah.
Oh.
This is the diabetics and scheduled for amputation.
Oh.
Diabetics healing is very slow, and with proper laying of fibroblast and other things which suture the wound together, that process is delayed. So what he's saying, probably that stimulating agent, it will act through the fibroblast only, but that process will increase.
Yeah. So we believe that even in very, very slow number of stem cells, they can able to participate such cases regenerations.
You use some in vitro culture.
Yeah. Before.
Products of in vitro culture, they may be applied.
Yeah. So it's just in vitro. We did a lot of work on in vitro to make a proof of concept, proof of concept that such factor has able to. And based on this, again, we need to make it person to person, it varies. So then again, we need to verify the patient's profiles, a lot of things. And then we can see, okay, this is perfectly fit for his skin. We did only in skin and so on spinal cord. This is also spinal cord. We have, before moving to this, we have treated 45 dogs, experiment model, and they're successful.
What is the mechanism of erythropoietin here for the local healing?
The concept is, if I say very simply, so we need a neurons, we need a blood vessels, we need a bridge. So if you try to connect, you need a mobile network because you need something from other part of the body. So neurons should be very functional. So any organs, let's say spinal cord, brain, kidney, they need support from other part of the body. They need to send a message to the brain. Okay, I am in urgent conditions, I am in very urgent, and please do something. Because they know better. We don't know what they're sending the message.
No, that's fine.
So we need to make neurons.
For erythropoiesis,
Erythropoietin is
on a select population of stem cells which are going to make your RBCs or red blood cells.
Yeah, this is also.
What I'm asking is in neural tissue, which is the counterpart? Where does erythropoietin work?
Yeah, erythropoietin protects. It's also regenerate the neurons. There are a lot of experimental study has done.
Mature neurons.
Yeah. arthropods and some other factors. Protect a lot of, I think, thousands of publications on that, but there is very few. Now concept is growth factors. It's not a common, so we need to make a clinical grade. So if you can buy the clinical grade growth factors, like then you can see that when you buy the clinical factor, any factors to make it injection in human, you can see one graphs for the supplier. So the mycoplasm and some other thing. So this should be still is a risk because all growth factors coming from the cell line. So it still is a risk. So that's the point. That's we need to choose a high quality clinical grade growth factors, and you need to see the graphs when you buy it. Some company are quite good, and now some recombinant researcher, they try to make it different way. Different way means there's few publications that let's say they make a peptide sequences of the growth factors. Now it is discovered, they try to find out, the peptide researcher try to find out that very specific sequences of this growth factor has activity part. So now they take out only some sequences of the growth factors. They don't want to take the whole factors. But it's still is, little research is done. Maybe in next 10 years we don't need the growth factors. We needed some sequences of the peptides. And let's try to find out the functional part of the growth factors. So vitamin C always good supplement, and then it's survived. And we have treated also in 900 horses in Dubai for tendon regenerations. So we apply the concept to the different organs.
The spinal cord injuries are.
Yeah, please.
Excuse me.
Please.
Spinal cord injuries can be treated with injection of stem cells also, na?
No. As I mentioned that we don't prepare to isolate the stem cells from body. We know the stem cells in the injured organs, and we try to target them. We need to make a special recipe, special food for stem cells. We need to make a special recipe and give to them so they can activate and give a superpower for regeneration. We don't take out the stem cells here. No. Stem cells are there, and we analyze the patients, and then we found the combination. This is published in. It's free. You can just put my name. You can find it. You can see the how characterized before and after the patient. This is the case studies. The one patient. So I appreciate a lot of doctors can also try. Yes.
So I have one question actually. You were saying that the same thing can be replicated in a solid organs as well, like in case of heart actually. It's a heterogeneous population of cells are there. So if you want to activate the stem cells locally.
Which organs?
Heart, I would say.
Heart. Okay.
Okay. So if you would like to activate in-house, locally, so how will you navigate those cells toward a different type of cells? Because it is more complicated as compared to any other type of.
As I mentioned, that's nobody knows how to navigate and how to work perfectly. But if you see, you make some in vitro models. Let's say you started experiment for cardiac progenitor cells, and we try to target some diseases. Then you develop some model, and you say in these combinations, you found the purification of stems, cardiac mitochondrial cells, and everything is gone. Very good. Okay, you optimize. And then when you try to inject them, if we try to understand how it is work, molecular mechanism, then we spend our whole life. So if we see these things, and then the heart can regenerate and heart getting good functions, there is a marker, then we can say, okay, so we target the stem cell because 12 or 13 publications, including us, in heart, I show you. So next 3 to 4 years, we try to inject in heart also, but we detect it. we compare the healthy heart, disease heart. So we know where it is, locally present, and then we see the function of the heart. And some cases you can take small part of the heart and then then you can analyze. That's the like histopathology, like here in the last slides in 12 markers. It's a perfect, excellent skin regenerations of the markers, and we found all positive. Not in that day 2, in 12 days, means around 2 weeks. We found everything perfect skin regeneration with all markers, like a growing skin, like children's.
So I have one more question actually.
Yeah.
Apart from that. So in case of the myocardial infarction, actually,
Yes.
we use quite a few cells, millions of cells, I guess, like when somebody is having a myocardial infarction. Now you got a very few cells, I would say the adult stem cells locally inside the heart actually. So I have a doubt about the number of cells they will generate and in limited time frame, because then you need to get those number of cells, or maybe you need to replenish the lost cells.
Yeah, there is like in New England Journals of Medicines, I think 2016, it is great work, and they proved also that bone marrow transplantations, like the old method, still working well. Still not bad, it's working good. But if you have a major problem like diabetics, that time bone marrow does not work. So we believe that even in small number, we don't need to put. Depends on the condition, it's difficult to say. That's how many number. If you put the large number of cells, maybe they will be cured. But we don't want to take from outside. Is the issue. And if somebody has a good regenerative power, and then maybe the bone marrow can supply, because they're moving faster than the rocket and to the heart, and they can. For example, in Nature Medicine, there is a very interesting article published that if some liver failure patients, if you can take them from hepatic circulations, you can find a lot of cells, stem cells, circulating around the liver. They try to help, because the neighbor organs, they try to repair the organs. They found a lot of cells. In general conditions, you cannot find such circulatory stem cells. Yes.
So I have a question, because you are talking about the local stem cells and making them activated by the specific cocktail of growth factors.
Yes.
So when you are injecting this local growth factor, how you are defining that you are really activating the local stem cell, because they should be automatically activated on injury.
Yeah. A lot of things. A lot of, like, thousands of mechanisms can go. We did it's a good question, but things is that it's just like we do in vitro works for cancer initiations.
But in vitro is completely different, sir.
I know. So as much as possible. So it's a thousand of things we don't know exactly. But we need to make some objective. Like a cancer initiation is a major problem. Cancer cells are moving. So we, like a lot of things, we cannot control anything. Anything can happen. We don't know. But we need to control in vitro model how to prevent the cancer initiation cells. When you develop this cocktail, we have one special experiment with cancer initiation experiment. If this make a little cancer initiation supports, then we don't support this combinations.
What kind of cancer initiation experiment you do, if I can ask?
We do when you make a lot of we make special combinations, and then we do cancer. We do some deep gene sequencing. That's deep gene sequencing in microarray. We see the all cancer profile. We have some software, but we need to just take a DNA and deep gene sequencing and PCR microarray. Then we see that this is have no cancer initiation cells, and we take a fibroblast, all type of different heart stem cells, and we learn this from in vivo. We don't know, we cannot do.
Yes, sir. Let me
Yes.
keep my point. In vitro is quite different when you are putting the
Culture.
Yes, totally different.
And in vivo, it takes years for the tumor initiation.
Yes.
You know that it's not the immediate process.
No, no, no.
The growth factor, and most of the growth factor, what you are trying to put there, like IL-6, if I am not wrong.
Yeah.
These all are also pro-inflammatory factors.
Yes.
Pro-inflammation, inflammation, and cancer has a strong link. So how you are taking away the risk of cancer initiation in the future?
Yes. Yeah. It's very important. We did such experiment. We don't put IL-6, but we did experiment that when little inflammation, very important questions, and for this point for these questions, like millions of euro is spending. I'm telling the same concept as salamander. So when, as I mentioned, stem cells are silent.
Not everywhere, sir. Let me clear, because
Listen. Yes, inflammation. If a little inflammation is extremely good for regeneration, little. When you can see the salamander, fast inflammation, when they knock down the inflammation, salamander also cannot regenerate. So we believe little injury or little inflammations. But in heart, even we did not do any experiment in human. We just detect the stem cells in heart. We never try in human. In future, we will try to do this. But it's true that it's not easy. Lot of things to do. But we can consider. But we see just like if something curable, some point, then we can say it works.
That I agree. If something is cured, and we don't have any doubt about the treatment.
Yeah.
But my question is, nowadays stem cell concept is not, it's again miserable little bit, because so far what we know from the solid organs, if you just forget about the blood, in the solid organ there is no hierarchy of stem cell anymore. They are hugely plastic. So any cells, or mostly differentiated cells can also revert back to stem cell.
Yeah.
So I'm little doubtful that all this cocktail is activating local stem cell, which should be anyway activated automatically.
I agree.
But they are acting probably on the more differentiated cell or more progenitor-like cells.
Actually, we don't know. We need to establish. A lot of people are, as I mentioned, in heart cases, I told you, only 12 publications you can see. Other thousand you can find it. So it's a good point, but it's very difficult to establish. Very difficult.
Well, then we need basic research for the regeneration and all this, how regeneration.
Some group are doing the same thing.
Yeah, because the gray area in the stem cell field is, according to me, that we don't know how the regeneration is happening. Regeneration is happening, stem cell is taking part, but not means they are the not only one who are taking part on regeneration. Because if you see, like, I'm coming from the intestinal research.
Okay.
LGR5, probably you heard that they are the major stem cell in intestine. But nothing happens if you kill stem cells there. I mean, regeneration is still happening there without LGR5, which is one of the most bona fide stem cell. So
because I met the professor from Netherlands. So here is constant injury, the intestine. So it's like a gate point. So evolution point of view, many researchers believe that is the main gate has adapted like liver. A lot of time before going to the heart, liver has a high regenerative powers. So like this, intestine also encounter a lot of problem in every minute. So they adapt and they develop the special type of stem cell naturally, evolutionary, and that's why LG5.
But even LGR5 is not solely responsible for the regeneration. So there are,
they need a support. I believe if we make a special if we can create a good microenvironment there, it can get it, which is people are trying to do it, but it's not easy to create. I just simply say the combination of growth factor, but in broadly, it's mainly you can create a regenerative microenvironment on the local organs. It means not only the growth factor, a lot of things can happen because we don't know. So That's why the proteomics we do, like many people, every experiment when they make it in vitro models, they used proteomics. Now they are saying the protein-protein interaction also is a point. So lot of people, mass spectrometry become very common now in all experiment, and which can analyze 30,000-40,000 protein, the proteomic changes about this, what is on the heart profiles or not. But we don't have any other method. The mass spectrometry is the only methods we can see the 30,000-40,000 proteomics profiles. So that's why the heart is not easy. And 2009, before that, nobody believed that heart is stem cells. The paper published in Science that from 2009 till now, you can find in human cases only 13 to 14 publications, including us. So no, not so easy, but depends, because it's also related to the fibrosis. Excess collagen replaced by the cardiac cyst is also another problem. We generally consider the fibrosis control, cancer initiation, and some other point, purifications, and then we do in animal model, and then we did not go to the heart in humans.
I think it's just a matter of time, means that more and more organ will be discovered for the stem cell. First, we will call them resident stem cell or local stem cell. Then there will be quiescent stem cell discovered, like in many tissues now. And then the next problem will be the plasticity, how we control that. Because
in animal model, it's quite good, that's decellularization and recellularization. That is quite good working. But in human,
because we need more research on human. Human was not easy to access for the research purpose.
Yeah, but here you can see this paper is first one in my boss name. It is the trachea. It is a tube-like structure. It's easy, like a pipe. And this is from dead woman, decellularized and recellularized stem cells and transplant, and this still is surviving in London. So, but it's a simple tube-like structure. But heart and others is very complex. But a lot of people are working a lot to make it successful for decellularizing, recellularization. If it is successful, then it will be great invention, of course. But I hope it will be in future.
Decellularization is the purification of extracellular matrix. So when you inject the extracellular matrix along with the insect's stem cell somewhere, extracellular matrix provide place to sit.
Yes.
So lots of signals comes from there.
And in this case, in the decellularization, you get the scaffold.
Yeah, just a prem.
The extracellular matrix.
Yeah.
Provide scaffold for the stem cell.
So in decellularization, like these cases, I show you. This is the cases. And the thing that's, it's a tube-like structure, and then you can decellularize, taking from dead body and decellularize and putting stem cells, making a functional organs and transplant. There are another questions coming up after transplant. It should be properly transplanted, so it will be completely fit with the. So that's why stem cell mobilization agent has to inject to then a local stem cell from other part of body, so that it can be perfectly restructured and it works. And then that's why. But it's tube structure. We need a support from other organs. We need to think what's going on with other local tissue cells. But it's in human, that's like in heart, when you compare the location of heart stem cells, one PhD student, she took 4 years. And the experiment was to detect the status of stem cells and the healthy heart and some disease conditions. Only very, very simple, but it took 4 years, because it's not easy to get a small part of the healthy heart. And so before that, the stem cells, this you can see, this is published in Frontiers in Genetics, and the first one, healthy heart, stem cells are sleeping. This is in all human. And when you give the myocarditis, CM stem cell try to activate. So we found big expressions on those conditions. And this is published in front of us in genetics. We published it. And you can see how many number of patients, age of patients, and the locations. Here, these are the locations that some, like the heart stem cell present. Maybe some other part of the heart, but nobody knows. And some people believe that the local fibroblast, the local fibroblast also has key role for regeneration. So you can see the locations: left ventricle and right ventricle, right atrium. That is the location. And not so much publication on the heart. We make a table also, and in these publications we mention that cardiac progenitor cells cases in myocarditis and cardiomyopathy, qualitative and quantitative, because here, like Professor Pinning, who got Nobel Prize in physics, he wants any tissue, so they are doing a lot of work for cancer. So if when you do histopathology, any markers, we cannot see we missed some of the image, like deep gene sequencing. When you do PCR, real-time PCR, you can miss many oncogenes, many genes. And when you can do deep gene sequencing, everything can be catched. So it's very important, it's very important to use sophisticated technology to detect the missing part. In some cases, if you do real-time PCR, you can miss some oncogene. When you do some experiment and when you make a deep gene sequencing, we found the oncogene expression of something. So we recommend, it is a very expensive experiment, but we recommend to make it proper optimizations. This is image analysis of heart stem cells. And other students also doing brain, kidney, and heart. You see the small red part is very, as I mentioned, that number is very low and sleeping and silent, and we need to target them whenever used. This is the concept. But we have not done anything in brain and kidney. We just basically focus on skin, and we're now planning to do in some heart model. This is some experiment in mice. And in bone, this is also some animal models of bone. We try the same concept with bone because it's hard tissue, but we need some support because bone is we need a mechanical strength. So the TCP cylinder, and then we do it. It also works. And when you make a skin in animal model, we can see some hair follicle regenerations. So then we believe that these combinations can be useful for hair regeneration in future. So you see here there is some receptor. So we have not done so much work on molecular mechanism, but we found some receptor that it work only when they have, and we established this mechanism for some growth factors. We also use topical applications, and sometimes we use infusion, like blood circulations in bone in animal model. And here you can see, as I mentioned, the new blood vessels. If you can create new blood vessels in anywhere, this is a requirement for all organ regenerations. And then you can regenerate any organs. And this is the simple concept, but very important. Creation of the new blood vessels, creation of the new functional neurons, and then you can able to regenerate any organs because they need support from other. You have to create new blood so they can bring a lot of regenerative factors from other part of the body. And the blood vessels should be very thin. We are in our life, if you can see the thickness measurement is very, very thin. So it's called, let's say, neovascular vascularizations, or new blood vessel formations. And whenever treatments, we take the small part and then we see, like here you can see, you cannot see the fine blood vessels, but here when we make it image analysis, then we find the number, you can count the number of how is vascularizations. So image analysis is very important for in if you can evaluate something. But it's very expensive also. So here is controlled, you don't see. So new blood vessel new vasculation is important for regenerations. And some models like histopathology, how it's for epidermis, dermis, and how it degenerates. So we found complete skin anatomy after treatments. Like this is the major markers for wound healing, but in clinical trials we consider more markers. So this is all about, I have told about that's focus on local stem cells. Like we need to know, and it person to person is very, because like everybody their own immune system. So some people has a high immune powers, somebody less. You have a low immunity. Same like regeneration power. So we need to taking care of our endogenous stem cell system. And epigenetic researchers told they found new great invention, like every regenerative power it can recorded on the epigenome. So organ regenerations definitely it's varies person to person to person. So we need to make a little break.
Someone is asking.
Okay, please.
Sir, my question is, what is the difference between the engraftment in a hematopoietic stem cell against the solid organ whenever we are transplanting the cells?
Can you repeat again, please?
The difference in engraftment of a hematopoietic stem cell against a solid organ.
So,
In this case, the cardiac condition or myocardial infarction.
yeah, as you mentioned, that there is some publication in New Journal of Medicine. It works in some cases, but if the heart is the major problem, then they need some support, some more different support, and we need to work on that. But it works. Did I answer correctly or anything? Can you speak loudly, please?
Trying to transplant the hematopoietic stem cells against a stem cell which is tissue-derived stem cells, wherever we are trying to place it in an organ for the resumption of function. Suppose
So
we have,
basically,
isolated the cardiac stem cells, and we are trying to make functional cardiac myocytes by transplanting into the cardiac, the organ. Then what is the difference in the engraftment? The engraftment obviously is better in hematopoietic stem cells, and we are in the formative stages as far as the microenvironment, or is it because of the immunosuppression?
no, microenvironment is important for any type of stem cells. It's very important. We need to control the local microenvironment, whatever you target the cells.
So if we, does the immunosuppression play a part as far as the tissue engraftment of stem cells is concerned?
It's as I mentioned, that's not so work had done on the particularly on the heart, and I believe that it's important on the microenvironment. Whatever the cells you can use, whatever the cells you use, it's important to control the microenvironment. And cardiac cells is much better, of course, but they have a telomere, they have a lifespan. So homeopathic stem cells is ready to they can also create cardiac cells based on the environment on the heart. We have to know about the heart condition. Accordingly, we can say which is better, which is not, or it can be or not.
So, what is your experience in the engraftment?
We have not done. Unfortunately, we have not done in human. We just detect. Yeah, we try to. We are doing now for in vitro models. In vitro models, we just simple concept cancer initiation, and we try to figure out the heart microenvironments, how it is going well, and then. But we recently started for this. But we completed the detection of the stem cell. We know where it is in different age groups, and now we need to develop some cocktails, and then maybe in next 3 to 4 years we can able to treat something.
Thank you, sir.
You're welcome.
I have a question again. So when you are saying again the tumor initiation, so how you are really controlling? Suppose one patient
Cancer initiation.
Yes.
Okay.
Let me finish my question because you have maybe some mutation, some benign mutation in the patient sample already, which you did not catch in RNA-seq or whatever.
No, it can be catched by deep gene sequencing.
No, but tissue is very heterogeneous, sir.
No. Okay, I understand. This is the same questions going on in Japan.
So if you are taking from, one biopsy from this side of the tissue and you have benign mutation on another side, you will not catch it in
No, it's true.
And then you are providing the inflammation to boost this particular benign mutation into acquiring more mutation due to inflammation, and then you have a risk of, how you will tackle this in the future.
Currently I don't know. I need to think about it. But mutation is like in Japan, the same, more or less same questions is like they are saying, okay, RPS has a mutations, but now generally mutation happens in every 2 hours, every 30 minutes.
It's a different mutation.
No, sometimes it depends. But it's, I don't know, like how to do particularly focus now skin. Maybe I hope some research that they do for control the mutations. We just simple do cancer initiations, just simple. That cancer initiation cells are circulating and we need to control them. But mutation is not easy to control, not easy to establish these things, even in deep gene sequencing.
You are saying cancer initiation cell.
Yes.
Is it different from cancer stem cell?
Yes, very different.
How?
Like cancer stem cells is aggressive stem cells. Okay, aggressive means high proliferation.
Stem cell?
Yeah, cancer stem cells is highly proliferation because out of control, like a cycle without break. You are just going and no break. We developed, if you try to make a break of the cancer stem cells, if you put the extract of embryonic stem cell, then cancer stem cells can become a normal stem cells. It means the extract of embryonic stem cells has some factors. We need to find out.
Sir, if they have already acquired the mutation, how they will revert back the mutation?
Which mutation? It's there are thousands of mutations.
No, the driver mutations like P53, KRAS or APC in this line. So how your embryonic stem cell cocktail, whatever, will revert back all those mutations in cancer stem cell? I don't see any concept behind that.
This paper I'm telling about is published in PNS, and it's very interesting paper. They think because this all mutations also with children, when a baby develops in the pregnancy, all going up and down. Like Siniamanaka, select 110 oncogenes. And then he found 4. So we don't know which going up and down, how to control. We cannot say. If we can say, then we can create a lot of good medicine for cancer. Not easy.
Did they discover any mechanism for that? For example, if you are making extract from embryonic stem cell and putting on the cancer stem cell and it converts back to the normal cell.
Yeah, because they thought, but when you see,
It is published.
yes.
It is published.
I guess
Then how it is
there's a confusion. Actually, it can't be embryonic stem cell. It could be a oocyte extract actually, because the oocyte extract in the oo plasma, those factors.
Yes. So that's why,
The non-good embryonic it should be the oocyte oo plasma, because whenever you do the cloning, what you do, you take the terminally differentiated cells, you put them back in a oocyte, integrated oocyte, and what they do, they revert it back into the embryonic state. The oo plasma those factors. The embryonic stem cells mean the first differentiation of the blastocyst, I mean, that takes place at the blastocyst stage when you get the ICM cells and the two-factor cells, right? ICM cells are the embryonic stem cells. Embryonic stem cell extract, I won't say that they have, you will take the extract and then they will put on.
they put the extract from embryo.
From the oo plasma. No, that's the problem. So, oocyte is having that extract, those factors. You know,
The PNS paper.
Got an experiment. So that's what he did in the tadpole experiment, right? So he took the terminally differentiated cell from the gut of the frog, then he put back into the integrated oocyte of the frog. That's how he got the cloned tadpoles, right? The oocyte has those factors. Oo plasma, the oocyte has those factors, which have the ability to reprogram the terminally differentiated cell back into the environment.
So we need to find out, like, thousands of factors are there, which we don't know.
It is very much doubtful that a particular mutation will be corrected, and then the cells will get converted.
No particular mutations. We don't know about mutations.
Cancer stem cell doesn't have any.
Cancer initiation cell. As I mentioned, that we do deep gene sequencing, and we have cancer profiles. Okay, so cancer initiation cells, there is a model. So if you put such things, like, and we in these conditions, cancer go making a cancer. We can make a high some profiles of the cancer. Okay, common cancer, particular for cancer initiation, which is circulating in our body all the time. Can make a cancer anytime. Depends upon the habit. And when you in positive control, negative control, we do, and when you see that in that conditions that we don't see significant less, when we don't see any in vitro model, I'm not in vivo, and then we believe that it is works. In skin cases, we did it. But mutation, you want to know the cancer profiles of the deep gene sequence.
I just wondering who you said that if you make an extract of the embryonic stem cell,
like a
cancer stem cell, then it converts into normal, right?
Yes.
That's what
Aggressive cancer stem cell to the normal stem cells.
So what is the property of cancer stem cell versus normal stem cell in those experiments?
There are a lot of publications. You can see that.
What do you consider that now they became the normal stem cell?
I believe all stem cells, even endogenous stem cells, bone marrow stem cell, cancer stem cells, they are 90% similar.
Yes. So what is that?
The thing is that we construct the break. When you make a differentiation, like we did a lot of work on iPS cells to differentiations for skin, liver, heart, and when you make it, you don't need to make a termination. Then they can go anytime, any cells. So we also work very carefully the termination. Oh, your work is finished. Now you have to stop. We need to develop some things to stop them. Otherwise, it's going like liver. We work for 10 years for liver. We want to know which transcription factor doing for regenerations for the liver. If we cut half of the liver, still we are not finding because we have 25,000 protein profile, and it's not easy to find which is good. Many people also working for that. So
this how transcription factor is identified in particular
mass spectrometry, and then you need to
just gene expression profile, because in a particular tissue the level of that particular transcription factor that drives towards a particular lineage is very high compared to the other type of this body cells. It's very high. For example, in the muscle, MyoD level is so high.
I understand. So, now people are using the placenta extract for skin regeneration. They know platelets. So, there are a lot of factors we don't know actually. We don't know what is there. If we catch them, like salamander, or people are doing a lot of money, but still they are not found. What's going on? Electrical signal, how the migration, everything. Still, they just do knockdown. But there is few interesting experiment that if they knock down some gene, then it cannot regenerate nicely. So they found some clue.
They are important.
Sorry?
That's why those.
Yeah, it's not easy. Not easy. You can see like very nicely limb generations.
Right now it's very easy. Now we have the library for the CRISPR, shRNA. You just screen yourself. You knock down all the genes.
Yes. Yeah, but CRISPR also you have. I think you have seen the one article from Germany. They have for skin transplant. And that's how they make it iPS cells, and then make it small, like holotypes. If you can, when you make it, when you create iPS cell, any stem cells, you can see some type of very special cells, and they isolate it and then transfer. This is published in Nature Medicines recently, I think 1.5 years. So they cover completely, like 80% of the whole body of the skin is recovered. They got a lot of money from next treatments. So I believe that stem cells can able to do, and they know how to control mutation. They know how to. We believe it's not easy to establish everything. We know that they can control mutation. They can control oncogene initiation. They can regenerate everything. These are usually master. So we need to activate them. We need to give them food. We need to supply something. And how they handle it, we don't know.
So regeneration and cancer initiation, tumor initiation, they share a lot of common paths.
Yes.
Okay, so do you think your treatment of this cytokine cocktail or whatever cocktail you are discovering, in the future, can you use them for the cancer treatment? Or
we believe it. We believe it.
Have you tried any?
No. We did some epigenetics concept because we believe that basically we combine now epigenetics with this. Okay, we know because we know some factors, some combinations. We try to make a structural part of this growth factors. Even we don't want to use the growth factor, we try to use some herbal things. So there are a lot of growth factors, like lot of herbal ingredients mimic the growth factors. There are a lot of research has done, and still we are trying to use this botanical supplement to replace the growth factors. So we combined this and we started some epigenetic research on that. So we tell to histone protein, please silence this gene. We tried we planning to do some in vitro knockdown cancer model using epigenetic approach by botanical extract and as well as this some combination.
What exactly you are planning in that epigenetic approach? So what you will be looking for actually?
We just we are looking for which cancer?
Very broad area. They have so many proteins doing modifications, all different type of modifications.
We make, let's say we make it for skin cancer or some. We can find. We have to target only 1 or 2 cancer.
No, it's okay. You are targeting the cancer, but I'm talking about what sort of epigenetic modification you will be testing or looking for.
We just very simple, like acuity model, like acuity mice, simple model. We make it high throughput screening, and we just see that this gene is silenced. That's all. We need to see. Okay, we run thousands combinations, and then we try to see this cancer there, but it is silenced. That's all.
You will do the deep means
We do, yeah.
high throughput chip analysis from those samples treated with your botanical
Yes.
extract.
Yes.
Then you identify.
Then we want to see that, okay, if not work, then we again the combinations, because it's a simple and it does not take so much time using but it's not easy, because just imagine, we try to replace the growth factors by botanical, which is many scientists they don't believe.
But you said you did not tested those growth factors so far for the animal model. So how?
No, we have done. We have done small animal model, large animal model.
Have you tried any of your cocktails in human, like in a trial, as a trial in a human, where you have seen something
Negative.
positive, like similar, like regeneration
in the tumor, a human.
Tumor? No.
Cancer.
No. We did a lot of time on only skin, few cases in spinal cord, and few cases on tendon. Mostly we spend on skin. Mostly skin. But we are planning to do in heart, because we found.
The experiment you were talking about, where you have used this embryonic extract and you are able to restore the cell.
No, this is just a take-home message that something there. We cannot do anything. Nobody can do anything with embryonic extract because it's ethical issue. Lot of complication. So this is just a scientific message that, okay, something is there, you need to find out. Like Shinya Manaka's concept. He made a review articles on Japanese language, on what switch on, up and down, and then he selected 100 something to make it go back to and then he's successful.
So I have a two question or comment.
Yeah, please.
So do you think it should be better to move rather than preventing or reverting back cancer stem cell into normal stem cell to make them terminally differentiated? Differentiation therapy is now in talks. So
Yes.
people are talking about differentiation
Yes.
therapy. So this is one of my comment, and I would like to hear your comment on that. Second thing, I forgot what I will ask.
Okay. Yeah, this is also when, like Shinya Manaka in from 2008, we spent a lot of time to use the not onco factors. We're using the chemical. You see one article in Science that chemical induced iPS cells. Then we need a lot of money, a lot of time on human, but it doesn't work. But and the students are frustrated. Oh, we spend 1 year and we don't get anything. Then we need to change the topic because they want PhD. So sometimes they are angry. So, and then I tell them that leave them plats, because 4-5 months, then finally you don't see any colonies. Many people like us, they try to make little modification and try to make it quick on human cells and then make it publish, the past was in Science. So, but nobody is success. It is.
Paper was retracted, I think. If we are.
That was from the Kuba cut.
That's I don't know, but it does not work.
The
The pH from Japan, that is a different one. But this yeah, so it work in mouse cells, but it doesn't work in human. But when I kept it for 6 months, we found some 1 or 2 colonies. But who will work on that now? We need money, we need manpower. But I believe but many companies spend a lot of money, but nobody works. But science, nobody does not know. But if chemical will come, then everything will be controllable. So
the chemical also we can induce differentiation. That was my point. Actually.
So terminations is even we successfully establish the termination in vitro, we cannot control in vivo.
It's the same.
The same.
The condition.
So all treatment has little risks, but we see the complications and inflammations and not. But still, it is work. Then we need to pass the regulatory limitations. Then it's possible. There is some like in Japan for iPS cells. They are doing a lot of work for control the mutation. There are a lot of argument is going on. And they said mutation, this is normal. We can see they take a healthy tissue, they saw the mutations. Like if you have today, next day, different mutations. So we believe that's cancer come from, we have cancer initiation cells, we have a papillomavirus, lot of things, we have retrovirus. So everything inside our bed, good things are inside our body. They can activate any region, any time, then can become a cancer or any diseases.
Have you done any correlation study with your regenerative models and microbiome? Because now we
No.
Because now we know that especially in the liver and intestine, they are heavily controlled by the microbiome.
Yeah, that's why we call two brand. This is one brand, this is one brand. They work together. And if you try to make a beautiful skin, then you need to take care of microbes. Then your skin will be beautiful. This is just we are giving teaching now, but we did not do any experiment. But we are planning to do some food supplement, food capsule. But we are apply for some funding, food like beauty capsule. So if you take one, because it's crazy markets, so you need a one capsule which can activate the your all friendly bacteria. There is the collagen, and there's the concept that is also the pomegranate. This natural medicine paper from Switzerland. So pomegranate, if you eat pomegranate, our microbes eat, then make anti-aging foreign. So this concept, but number of the cells in microbes is higher than the number of cells in our body. So still we try to find out, because the food supplement cosmetic markets is big markets. So if you can develop something, some capsule, and then it's easy to get money and you can establish a lot of things. So that's why I did a lot of survey work, what have done for the last 10 years. And then we found and we design, we write proposals and submit it to some investor, and they agreed to give. But we don't have any more experience on that, and that's why we try to collaborate some things. But for botanical supplement, we believe that it will work, which have done on stem cells, and we try to use this formula in stem on microbes. Let's see what's happened. If not, then we need to do this.
One of my last comment, when you because you said you are going to use cardial animal model for the animal models for this cocktail.
Mm-hmm.
So because, and you also said these resident stem cells are very few in numbers, right? So can't you use animal models with this linear spacing model, and then to demonstrate? This is just comment for your experiment. Demonstrate if really you are activating stem cells or you are inducing de-differentiation.
Anything can happen. We don't know.
So, but that
we have not decided. We have not decided yet that we are going to the animal model for this, or we can try some patients which are almost very complications, and we see. We not decided which animal model, but what we can do, we can take a histopathology, and then we see some of the objectives.
That doesn't tell if it's the regeneration coming from the primary stem cell, or due to the de-differentiation, or due to the quiescent stem cell. It doesn't tell anything.
Yeah, no. And basically we focus for the fibrosis, cancer, and good microenvironment, and for some markers, special markers. If we found strong, then we believe that okay, this is okay, and then we jump to the human, like in Germany for 9%, not on the clinical trials. So
Academic trials.
academic trials, and then we can ask for funding, because if it develops a concept is okay, then second point is that you need funding, which is not easy. Like microbes, if you write a proposals, some of my friend, it's very interesting topic, very interesting topics. So it's how to get fund, and some agencies they refuse to give the fund for microbes, to do. But now government knows that it's very important, and a lot of publications coming from last 3 years, very interesting article. So now the politicians also know, okay, we need to spend some money, like salamander. So now they decided to do some money on epigenetic and microbes. So now that's why we're adding also some grants on that. But you can see from last, before 5 years, it is not so many. But now it's very interesting article coming up day by day.
Every edition you find.
And very interesting one. So it's like a science, sometimes science are just sticking in one point, then like cancer get point, which is the Japanese Nobel Prize. He said sometimes some scientists just follow each other, but I believe that it should be like very important to do research on the microbes, because this is control. If you can taking care of your microbes, half of the disease is go away. We believe it. Because a part of the endogenous stem cells, we have a good database. We have documented, particularly I documented more than 1,000 people which have a good lifestyle. Like in Germany, there is an institute called Life, and this institute has been established by some concept that some people 35 years old, they don't have any disease, looks good, healthy, but they're dying by heart attack. But some people smoking 2-3 times and they are quite good. Even they are going by bicycle, climbing the mountain. Then we need to know what is the difference, because some people are looking super healthy, but they are dying by heart attack. So finally, there is some survey, because in survey reports that's okay. Because of our lifestyles, that's the fibrosis happen, like excess collagen synthesis replaced by the cardiomyocytes and heart pumping anytime because of the lifestyle, and this is a very big problems. So that's why we also make a preventive medicine, how we prevent the diseases. Like even Lancet, new medicines, they make also meta-analysis. If they're eating some fruits, yogurt, how they make it, there is a publication in the heart about the heart problems. So those people who eat the fruits and some exercise, they make a better health, improve the heart. Other no. So a lot of lifestyle cases are coming like microbes day by day now, and we have documented that if somebody lived for 85, 95 years old, then we have some questionnaire. That's like my neighbor in Germany. She is 98 years old. Her daughter is 52, and her daughter has a heart problem. So we have some questionnaire, how we react. Then we found that happiness is the best medicine. Happiness is the best medicine based on our lifestyle case studies, but it's not easy. So, in fact, that's why to be happy, to make comedy, to make laughing is important also for health, like cytokines profile, a lot of things for to make it health. So this is the lifestyle. It's part of the medical tourism also. Some patients are here for treatments. If they move to the another places, they improve. And now it's become a medical tourism. So that's why German patients coming to the Phuket, Thailand, there are a lot of clinic, like they're staying 2 months. They improve their diseases, diabetes, all of diseases. Indian, they are going to Switzerland, Germany, or exchange. So people, that's part of the medical tourism. It's a big markets, because every people, those who have a huge amount of money, they want natural regeneration, natural therapy, natural anti-aging. So they want to go holidays and they try to improve their health. It's called whole body rejuvenations or some part of this. For example, some people they hate to walk. If you can go to other places, if 35, 40 people are, let's say 5 people are not hate to walk and 20 people's interest to work, if they combine and they make an environment, okay, let's walk for 5 kilometers. Then the people that hate to walk, they can walk. Because it's like a creation of good environments. Like you have seen the movie in Munaba MBBS, last moment, how the father of the doctor was cured, so it's true. So a lot of cases happened like this, like happiness, social things like stroke and a lot of depression, a lot of diseases has been cured by medical tourism. Just moving one place to another places and making some homely environments and family environments with new people and something like that. Actually, heart is not easy, so we just consider the fibrosis because fibrosis is important part of the heart attack, even in knee. So excess collagen, excess fibroblast is problems in anywhere. So these things and cancer initiation normal. We just use microRNA deep gene sequencing, and then we see it works. And we prepare to use some of the growth factor which is regulatory approved. And now we are trying to mimic, we try to find out other replacement, which is not easy at all. But if you find some growth factors and then I can tell you, okay, this botanical supplement is mimic this factor, which is not easy. So we need to find out how it works and how the functional points, and then let's see how the experiment is going on. So this is all about the endogenous stem cells, how we can part and what is the status and what we do. Particularly we have spent a lot of time on skin, and now maybe I can tell more about the iPS cells which you have done. You may have had 2 grants from European Commissions. So we have a little breaks or any questions from which?
So what are the major pathways activated by this cocktail in a stem cell? Do you know some of those?
Very few. In particular on the skin, like TGF-beta 3, TGF-beta 1, and TGF-beta 2. They are 99% similar. TGF-beta 3, 2 and 1. So if we can develop anything which can stimulate the TGF-beta 3, then you can use for skin regenerations. The
cost of tumor initiation.
See, one company in London, listen again, I repeat, TGF-beta 1, TGF-beta 2 and 3. TGF-beta 3 is important for scar-free regenerations. When you see in embryonic conditions and baby wounded, then it can be repaired automatically because of the high secretion of TGF-beta 3. If you knock down TGF-beta 1 and beta 2, TGF-beta 3 cannot participate nicely. It means enemy should be, little problems should be there so that everything will be okay. You understand? Like stem cells, little inflammation is required, Here, TJ beta 2 and 3 has a different role, but their presence is important. TJ beta 3. And then this London company, they believe we can make a TJ beta 3 recombinant proteins and making a skin. This company collapsed.
Beta has been shown for the immune suppression in many tumor entities.
Yeah, let me complete. So this TJ beta 3, they create the TJ beta 3 recombinant proteins and they use these things scarless, it does not work. You can ask me why. What do you think? TJ beta 1, 2, 3, this is the simple scientific floor beyond. And then this company make a TJ beta 3 recombinant proteins. When they use, it's failed. It's like phase 2 clinical trials failed, so it's closed. They lost a lot of huge of money. It means, I believe, that's TJ beta, it's important to be present TJ beta 1, 2. Like, as I mentioned, that's for microbes, we are writing projects, so I have all documentation to write proposals. Now I know what's going on. And when I give this lecture, microbes and beauty in Russia, and then one student asked me, that's okay, it's good to activate the good microbes, but there is some microbes are not helpful, but still there. Then why they are there? I said, I don't know. We need to find out. Like if you have some enemy is there, then our defense will be strong or something like that, So if it's nothing, then everything will be safe. So I think there are some always little problems there, then it will be high, everything will be well established, I believe, if I say simply like this. So this is thing. So we have our friendly bacteria, unfriendly microbes in our intestine. So if we remove them, the student asked me, if we remove because unfriendly bacteria, we don't need them. They are just coming there. If the friendly bacteria number is going down, then unfriendly is going up. Then we make a problem. Why should not remove them all them? Why not kill them? I said, I believe that they are naturally present means we need to control them. We don't need to remove them. This is nature. We don't know. A lot of things we don't know. And what do you think? Everybody knows. Some example that you see here, like glycogen storage or alpha antitrypsin deficiency or Wilson diseases. So these all syndromes, and this is called like rare diseases, and it's not easy to develop some medicines for using the conventional drug research process. So in these cases, the IPS technology has great role, because if you want to develop some medicines or try to cure some diseases, then you can take the skin cells from some disease patient and you can display this pathology, and then you can throw out screening and to cure this pathophysiology problems. Then you can say, okay, this works. So here, when you take a skin fibroblast from 10 years old boys, and within 23 days you see iPS cells, and when you take a skin cells from 72, then it takes more times. This is also some epigenetic roles. But when you take a skin cells from 10 years old boys or below 20, then you can get a high chances of iPS colony. And then because when you see the growing children or under 20 take a skin, they secrete a lot of different factors. In all, they don't. So this also has some supplementary role for creation of iPS cells. So the concept is that skin, we have to make it any type of disease cells below 20-25, because skin cells is very little, so we don't need to make any big biopsy. So then we can make a high, good quality of liver cell, heart cells, any cells, then kept it for your future therapy. So
OCT4 is a nuclear transcription factor, right?
3, nano, FOX2 and OCT4.
OCT4. But why it looks, the green color in the third slide looks like outside the nucleus, right?
It's a confocal.
Yes.
So you mean the DAPI and
Green color is.
you mean the DAPI nucleus and why it is outside the nucleus?
Green color is OCT4, right?
Yes.
But it looks outside the nucleus.
Yeah, I'm telling this is the thing. So during confocal microscopy, you see maybe there is some DAPI. There is some. You can see the right corner. You see the DAPI and cells are in the high magnifications. You mean like in confocal microscopy you can see like this. It's not that only then you can make a magnification. Then if you want to look at the OCT4 is only present inside the nucleus or something like that, then you need to make a different magnification. But this is some slides we have more. This is published in Stem Cell Research. You can see other slides. And we use the epigenetic process because epigenetic process has a low oncogene factor. And we make it iPS-derived hepatocyte cells. So this albumin, HNF4 alpha, and CK18. But we found that double nucleus in the right. What happened? Somebody calling?
Also, like these are good.
Okay, so you see that when you make it skin cell or iPS cells and then hepatocyte cells, it's not so easy to see the double nucleus of hepatocyte cells, but we found many of them, let's say 15 to 20% of the each plates, we found double nucleus hepatocyte cells. So it means the differentiation process is quite good, but not high efficiency because hepatocyte differentiation is still in the process, but we try to develop the efficiency process.
This is human iPSC?
Yeah.
What kind of culture conditions you have used? Because I don't see any feeder layer, so it's a metrigen. So you have you're culturing on the metrigen.
This one.
Yeah. So
This was
in feeder.
The epigemal was in metrigal, but this one is peptides. This is without matrigel, the minicircle iPS cells. This is will be online soon. So we make some peptides. We put this and it's growing, but otherwise in matrigel. It's a standard procedure which is most efficient for. But once it's become hepatocyte cells, we need to remove them from them because hepatocyte cells needs different support. If we keep them, these hepatocyte cells in this matrix, then it can be go to another cells or anything can happen. So once we got the desired cells, we have to move them out and then create the new environment, new in vitro bioreactor. This is the published. You can see this is one. Yeah, you see here, like this is iPS technology. Means we make a cells, whatever is liver cells, heart cells. Once cells have, then next question, how we need to keep them healthy. To termination difference is okay, and we need to make a purification. We need to see how much percentage of cells, because heterozygous population, it's not 100% efficiency. All cells are not hepatocyte cells. Maybe 20% or 25%, 30% are hepatocytes, other 70% cells, we don't know. So that's why we need to sort it out with sorting machine, Miltini machine, and then we need to take out, and then we need to culture. We can make them for more number of cells. Once we take out from the cells, and then we need to make it proper. If it's the liver, then we have to mimic the liver conditions. So when you get liver cells, the second one, the B, plates, is not mimic the in vivo conditions. So that's why you put the hepatocyte cells in the middle, the cells will die in 3 days. So we optimize, we develop this bioreactor which is called, we call bioartificial liver, and this is mimic the C1, our bioreactor is completely the physiological matters mimic to the liver. So we have different type of bioreactor for different organs. So 2 points. One is the liver hepatocytes consume 10 times more oxygen than the other mammalian cells. So that's why this bioreactor has both type oxygenations. So in bottoms, in plates, you can make a thickness. So they cannot get oxygen from bottom. They can get oxygen from other upper part, even is a medium there. So they are not getting, they're dying. So in a bioreactor, cells are survive more than 3 to 4 months. And you can see the differences, the oxygenations in human liver is, see, 2000 moles per ml, and in bioreactor is the same. And the distance also is around 20 micrometers, and in vivo there, but in middle one, you can see 150 to 200. So that's one Nature publication in 1965 that oxygen requirement of the liver cell in vitro. So that's why cells are dying. So once we develop any cells, then we need to take care of them properly. We need to make them healthy so that we can run a lot of experiment. This liver. And we have different type of high scale models. So we have large bioreactor, and you can see some other, this is all published in biomaterials. So we also differentiate directly here iPS cells. How, like oxygen, like and formula, like a flow model. Flow model means in static culture here, in the plate, in the B, cell metabolized waste accumulate. So if you try to screen some drugs or something, they make interference. So that's why not recommended. That's why in market a lot of more than 1,000 drugs has liver problems. They're rejected from markets. So in every drug testing, we need to make a hepatotoxicity testing using the devices. And here everything is well, like try to mimic the human liver. And this big model bioreactor we try in the pig model. This is also will be online soon in scientific reports. And pig also survived in pig model. The intercellular pressures and all ammonia regular efficiency in the bioreactor. This last one, and you can see that survived. So Pika survived with these supports, but without bioreactors, Pika died. We did it in medical school with Hanover. Yeah, this is. We also try to make some epigenetic models. That is DNA is not our destiny. It is true, and mutations happens anytime, and our DNA is changing, and we need to think about how its effect for regenerations, particularly for activation of stem cells. Like all cells in our body have same quality and quantity of DNA, but it need to be expressed in specific location and specific places. But eye formation also present there, or not present there. So anything, you can create any organs and any part of the body in principle, because the all cells body has. But we have naturally, they know which organs should be which location and which time. This is nature. So they know better how to generate organisms. So histone protein and DNA. Previously it was thinking that histone protein was not just present, just calling around the DNA, but it is a lot of evidence coming day by day that if we try to control the DNA, if we try to switch off any DNA, then you need to go through the histone is better without cutting and defect gene and putting a new gene, which gene therapy is failed. So histone protein, if we try to switch off any bad gene, you need to go through the histone protein, and you need to find out which things need to silence this gene, then it's fine. Defect gene is there, but no expression is okay. So the 6 epigenetic medicines has been approved by FDA, and it work very perfect, very successful. Like patients are getting a lot of satisfactions. Yeah, so this is the model which is new, and writer, readers, and writer reader. So, but the new, some epigenetic models many people have done, but very new things is that if you have a in the DNA.
Skin for regeneration, you have been trying with diabetic foot. So for liver, has any such initiatives been taken? Like for hepatitis?
Can you little volume, please?
Yeah. Good afternoon. As in cases of skin, there
Yes.
have been diabetic foot regeneration that you have successfully tried. So for similarly, for hepatobiliary system, any of the disorders like hepatitis, has these techniques been used?
In our cases,
Yes, sir.
no. We don't see any complication. Even we don't see any scar. We don't see any complication in our cases.
So at the moment, the focus is to fine-tune the process instead of therapies for individual diseases.
Can you understand what this is? I cannot understand properly. Please elaborate.
So at the moment,
Yes.
we are focusing on figuring out the various stem cells that are there
Yes.
and where they are acting instead of trying to treat specific diseases.
Yes.
Okay, thank you.
So we basically figure, like it has been done. Actually, the local progenitor cells has a less chances of cancer, less chance of high power regeneration. So everybody knows now. It's proved much better than the bone marrow stem cell. So everybody, but it's not easy to isolate, na? It's very low number. So, but it is it will work very nicely, very successfully to work.
What do you think about organoid technology? You need only one cell to establish the organoid. And
Organoid technology, we did a great work. This is also from Netherlands. So organoid, you see, this is also extract, material extract. We developed the stem cell extract organoid, but we did not continue because we focus. And when I speak, tell them that we can, they say it will be great. So I can send some slides. That's like organoid means like all cells are holding each other, then they work very nice because they exchange the molecules, which is very cell to cell signaling. Then you can see good. Of course, the organoid mode for preclinical research is quite appreciated because you can see the better pathology. It is a great model, but still it is from purified extract from material. But it works.
no, but you are purifying cells at least. So the limitation, what you are saying, to isolate a stem cell from the body, I think that we have already passed. Now we have to establish how to make them without the material.
Yes.
But we can grow means the low number of stem cell from the human body into material as an organoid, at least.
But you see the slides. The low number means very, very low.
We need one cell, sir, for organoid.
But taking the one cells is also not easy. Not easy. Because you need, let's say, isolation precision. Okay, we know the location in the heart. We know in heart. And how we can take it out?
You don't need to take out this one single cell. You need a small piece of tissue, and then you can make them as a single cell, put them in material. You need to find a condition for each and every person, but it's possible, I think, because all the organoid system, like in liver, in pancreas, in intestine, in lung, in brain, they have been developed for human as well.
Yeah, they have developed, but we know the location. How we can take it out? It's not like once we take out some part of the organs, we have to sort it out. Otherwise, it makes heterogeneous. We need to make it.
Other cells will be died anyway. The differentiated cell will not grow in that condition. So you have to find a condition specifically.
Yeah, but it's not so easy.
It's not easy, but it's not.
People's stem cell funding is going up. Maybe one day it come up. So here is. It's very interesting now in Germany, and there is epigenetic memory called, and it is very few people have done some case studies. So you see, as I mentioned, that if you have a good lifestyle, cancer prevention, organ regeneration, anything, then it can be recorded there. In histone and DNA, it is a memory box. So, and it can be inherited up to 3 generations. So, if you have a good memory box on regeneration prevention, and your children and grandchildren has some bad habits, and then because of your strong regeneration prevention memory on epigenome, they can protect because of ER. It is totally new. There are not so many methods established. But in Germany, there are projects going on for pregnant women, how it is going on, population studies, but I have seen some cases. It's really very interesting, and like frontiers in pharmacology, they invited me to write an article on epigenetic memory. So it will be few months, it will be coming in epigenetic memory box, because I need put more cases. Epigenetic memory box, so it is very important. Another thing is that in some population health studies, it is.
What did you say about 3 generations?
3 generations mean you, your children, and grandchildren.
Memory is
Recorded, and it can be transmitted up to 3 generations, maybe more. But right now, it has been shown that it can be.
Like inside the cell, when cell divides.
No, like prevention, any all depends upon the lifestyle.
Is there any,
Even bad, even lifestyle, even some character.
is there any publication on that?
No, only case studies. Some done. That's why I'm writing this article from last 2 years, but it's not finished yet because I'm waiting for some studies. So even if you can see, epigenetic researchers believe that you can see some in diabetic patient, they have a desire to eat more sugar. If you can ask them, they know sugar is not good for them. They know that not, but still they have internal desire to eat sugar. Addiction also here. So now there is some objective how epigenetics, because it's important to know. So there's not so much method in Mensch. In Germany they have some special training on that. So that is that many people target to the histone protein to switch off the bad gene or activate the good gene. It's easy. Easy means it's not like a previous method. You know the model. You have to decide which disease you want to switch off, or you want to know which targets. You make a screening model. Then you have to show, okay, this is a control model. This is positive, negative control, and this is we successfully control these things, and then we can make a good patent. And that is we have to do. Easiest way, not so easy, but this is the way we can make it. Like kurkuma, haldi, जो हम हल्दी बोलते हैं. So, do you know that in Germany, like if you want to go to the markets to buy the kurkuma powder, you cannot find it because people are buying a lot. Now they are buying the kurkuma extract, not the powder. There was buying before because there is a small experiment has done. They found that the kurkuma extract, like a haldi extract, is 35% more efficiency than the powder. So now people buying the extract, and small bottle cost 35 euro, which we used to eat kurkuma extract every day during curry. You know, previously we can ask like my mother in village.
What is the benefit of curcumin in terms of epigenetics?
Yes, many articles. No, epigenetics I don't know, but there is some others. But people's getting benefit day by day for anti-aging, for skin. That's why they are buying. That's why the selling is like this. The extract.
To say that the curcumin is what functioning as an anti-aging agent.
Yes, of course. That's why.
And how it's working?
I don't know. I have not done, but it comes under food supplement. And when it is proved that extract, like when you see 10-20 years before, like generally they use the kurkuma with extract, but now everybody more can put powder. But now we can go back again in Germany. In many people now do extract because it's 35% more efficiency, anti-cancer, anti-aging. It's a great benefit for inflammation, anything. So that's why the company is selling now extract. So many people also working for how to control kurkuma and some others. There is some very many other herbal capsule extract also. So now is the questions that's powder or extract, which is better? And I believe the extract is always better. Powder is purely purified, like going to the battlefield only king or king with soldier, which is better. So extract is complete frame of the soldier, king, and everything. So powder is something. So this like for a classic example. So extract is much better. People are getting benefit. That's why they are buying. And all healthy expert from India. Mostly India. But I ask them when I see them, it's a lot of people are buying. And we have to follow this extract model in our curry powder. Like we have to put them. It will be much better for us. Many it works nicely, and many people has been cured for some skin problems. So that's why they are buying. Even it's very expensive. A small bottle, €35 means it's very expensive. And we have here very low expensive. So the lifestyles also, and there another interesting example is that some people, like around, if you can say, okay, at 20 to 30 years old, I don't sleep well, I go to the party, I don't sleep 2-3 hours, McDonald's, fast food, but my skin is quite good, and I'm okay, and I'm healthy. But epigenetics says, no, your bad memories is recorded there, and it does not mean that it can express now. And then after 30 or 10 years has a good lifestyles and they have problems. Then they say, I am vegetarian, I sleep well, but why we have such problems? So epigenetics says we need to analyze not 10 years, whole life. That's why we have make a questionnaire from childhood to till now. And then that can be activated at that time. So many people, like around several hundred of cases, we have also documented the same things is true. And some of them have the like 15 years of the good lifestyles, now they have a cancer, highly aggressive cancer. 15 years of the vegetarian, good food, yoga, exercise. But 15 to 25, they have a very sleeping 1 hour, 2 hours. Because it's a question mark, some people has a unexpected cancer, even they are doctors. So it means that's why epigenetics make another objective for epigenetic memories about this. It is not well established, it's just now documentations are why happen and some little experiment going. So it is believed, and it is some scientific proof that it can be recorded there and it can be activated in future. So we have to taking a good memory box there for us, for our children and for our grandchildren, health-wise. So this epigenetic memory will frontiers in pharmacology. It's coming. Very interesting example with picture. I think it will take another 6 months. You can see it. I am also guest editor there, so you can. I will let you know when it's published. So people now jumping for molecular research to target the histone protein to switch off the bad gene and activate the good gene, particular for anti-aging and these things. So they move target for the molecular DNA to the histones. So that's why throughout the year, lots of training going on for epigenetic models. Because you need to find out which combination, which herbal extract, which things can tell to the histone, and histone will tell to the DNA, stop it. Your presence is okay, but don't express. Then problem solved. Any enemy is here, but they don't harm us, it's okay. We don't need to cut the bad things and move out. No, then a problem. That's why gene therapy failed. So acuity mice is a famous article, that's how that becomes. And Dolly was believed that it was not epigenetically cloned. That's why. But now in China there is 2 monkey came up. We have some documentations of the smoking habits. Some people have a 5 years smoking habit, then they reduce for 20 years, but they have a cancer. So it is recorded on memory box. We believe, but few experiment is still going on. Some other we have not started yet. And this is like a bone. So when you make a knee models, here you can like tendon, how it's going, flow models. Then we put stem cells, this is in vitro models, flow model, how the flow rates and how stem cells, I could consider embryonic development, we consider how the regeneration happens, then we develop dishes, not the plates, and then cells are quite okay for chondrocyte generations. And here just to see the circulation. So if any cell culture, they remove from one part to another part. So they remove the waste material of the cells and then give in the fresh medium. So like a flow model, like a blood circulations. So static model we not recommendable because static model always accumulate the waste materials of the cells.
Kind of continuous culture.
Yeah, like our body is blood flow is going on, and how we can tell culture static model is mimic the liver.
This is basically machine.
This is a bioreactor, small models bioreactor, and then this is also for chondrocytes. So from chondrocytes, we need to make its cytokine growth factors putting, and then try to make a proper chondro structure. Then when we put it here, then we found highly functional chondrocytes, and then with this thing we transplant to animal world is very well, and then if plates not going well.
You're showing it in epigenetics continuation. Is it correlated with epigenetics somehow?
Yeah, we started now that we try to find out how epigenetically control. So we try to make efficiency. All differentiation has a problem is low efficiency. And some epigenetic researchers suggest us that if we try to target epigenetically, maybe they make a high efficiency. So epigenetic experiments are like a ready-made model, and just when you have money, then we can get it. So that's why we try to do and devices. This is like decellerations. And this is this is for deceleration. You see this is the heart valve, and this is published in circulation at all. Here, see. Yeah. And here, and the transplant, and then the defect heart valve has been cured in SIB model. So, but valve and tube model is okay, but yeah, some. Then we also take some tissue and see the structure or pathology, and we also see the neuron cells or some other cells that are moving or not, biomigrate, number of purification, automatically it can graft. Yeah, this is our engineering part of the bioreactors. We can see everything, the flow, how flow, we can control, we can optimize. Everything using it's connected with computer, and sometimes we put the incubators. The incubator is especially for bioreactor. Yeah, and this is we in animal models. Right after transplant, the knee model, we make it grilling with a defect model, and then we transplant in animal model. And its little structure is properly well structured. This is amazing cells. Some slides are there, and this is we have most of them are surgeons, and he is my boss and secretary. I would like to thank you all of our teams. This is our building, and the last one is Bruno. He is a good friend of mine. He is vascular surgeons, and others, my PhD students, technicians, and right side is my boss. Thank you very much. This is the part of the IPS differentiations.